Hello everyone, I hope all of you are having a good day. My name is Darren Canon, a representative from Team C in our biology class, and I am here to welcome all of you in the Quarantine Survivor's Guide on Biology 101. So without further ado, let us all start. So on our last topic, as you all know, we all discussed about the cell and its structure. So for today, we will be discussing about how these cells divide in a process called cell division. Now, cell division has two phases, mitosis and meiosis. In mitosis, it only tackles on the division of body cells, while in meiosis, it tackles on the division of reproductive cells. With the two types of cell division, we will only be tackling with one, which is meiosis. Now, what is meiosis? Meiosis is a process where a single cell divides twice to produce four cells containing half the original amount of genetic information. These cells in meiosis are which I mentioned earlier, which is the sex cells, the sperm cells in males, and egg cells in females. Meiosis is a two-phase process wherein the two phases are Meiosis I and Meiosis II. Meiosis I is the reduction division, and Meiosis II is similar to mitosis, wherein sister chromatids are being separated. The goal of Meiosis I and Meiosis II are different. In Meiosis I, its goal is to separate homologous chromosomes, while in Meiosis II, its goal is to separate sister chromatids. In Meiosis II, there are no DNA being duplicated as in prophase I of Meiosis I. Now, you might all be wondering, what are the contributions of sexual reproductive processes to the adaptive genetic variability of an infant? So I'm going to answer that question. Independent assortment, crossing over, and random fertilization in meiosis introduces genetic variability into the new offspring. Gametes, as we all know, are the result of an immense number of genetic possibilities created during independent assortment and crossing over. Because of independent assortment, there are 8.4 million possible gametes that may be created even if crossing over does not occur. So, with that overview being said about meiosis, you now have a shallow information about the cell division. Now let's go deeper and know the different phases that happens during this cell division. So, you might want to read this book to know about that. So here, please grab it and know about the different phases of meiosis.
Wow, I'm sure there's a lot to know about meiosis from the Survivor's Guide. Why don't we start learning about the first phase, the prophase I? Prophase I is the longest, most complex, and most tedious part of meiosis. It has 5 substages: the leptonema, zygonema, pachynemia, diptonema, and the diakinesis. 3 main events happen during this phase. First is the condensing and coiling of chromatin into chromosomes, where chromosomes start to become visible as 2 fine threads under the light of a microscope. Second is the synapsis, where homologous chromosomes come together to form a tetrad. Homologous chromosomes are pairs of maternal and paternal chromosomes that are approximately the same size and have corresponding genetic sequences. Last, but certainly not the least, is the crossing over between the genetic information between the synapsed chromosomes. This is the most striking part of prophase I. Crossing over is sort of like a way in which chromosomes can exchange genetic information between each other. Each chromosome is now different from its parent chromosome, but contains the same amount of genetic material. In the 5th and last stage of prophase I, the diakinesis, the nuclear envelope has completely disintegrated. Meiotic spindle fibers start to form in preparation for the second phase of meiosis. The next phase is called the metaphase I. It starts when the pairs of homologous chromosomes move to the center of the cell. They orient themselves along an equatorial plane, forming the so-called metaphase plate. The chromosomes remain at the center of the cell until the homologous pairs are ready to move away from each other. Due to the previous recombination and crossovers, the 4 chromatids that make up each pair of homologous chromosomes are genetically dissimilar at this point. The spindle fibers attach to the kinetochores of the centromere, preparing the chromosomal pair to be separated during the next phase, anaphase I. But what does happen during anaphase 1? How about I lend the book to Shai so she can tell us a thing or two about the next phase? Here, Shai.
Thank you, Vea. And to answer her question, during anaphase 1, the homologous chromosomes separate and move towards the opposite poles of the cell while being guided by the spindle fibers. However, sister chromatids remain attached at their centromeres and they move as a unit towards the same pole. This is because the cohesins around the centromere remain protected by a protein known as sugoshin, preventing the sister chromatids from separating while the homologs are segregated. In the next phase, telophase 1, the sister chromatids arrive at the poles of the cell and begin to decondense. A nuclear envelope forms around each nucleus and the cytoplasm is divided by a cleavage furrow. This process is called cytokinesis. The result is two haploid cells. Each cell contains one duplicated copy of each homologous chromosome pair. With that, the 4 phases of Meiosis I come to an end. As for Meiosis II though, I'm not too sure. Why don't Christine take over? Here, Kat!
I'll take it from here, Shai. I'll be explaining the first part of Meiosis II, which is prophase 2. This stage is much more uneventful compared to prophase 1 because it lacks the exciting part of crossing over and exchanging of genetic material. I'll explain what I mean in a few seconds by showing you the model that we made. This is the model for prophase 2. Let me show you the chromosomes. Well, this one is the nuclear envelope, and this is the spindle fibers. As you can see here, the chromosomes further condense and the nuclear envelope disintegrates while the spindle fibers begin to reconstruct. That's everything that happens in prophase 2. Now that we're done with prophase 2, let's move on to metaphase 2. I'll show you the model again. This is the model for metaphase 2. In this phase, the chromosomes are again lined up at the metaphase plate individually as in mitosis. They are lined up in the middle, but this time there are no homologous chromosomes to be paired with. They are aligned through the facilitation of the spindle fibers. Spindle fibers are now attached to the 2 kinetochores contained in the centromere of each chromosome. Similarly to the mitotic phase, the 2 kinetochores are bound to the spindle fibers from the opposite poles, and they lie on the equatorial plane readying for the chromosomal movement in anaphase 2. And that's it for me. I'll pass this most helpful survival guide to Jazel. Let's listen to her explain the last 2 phases, shall we?
You're probably thinking how meiosis ends. There's only 2 stages left, and the first one is anaphase 2, which is similar to anaphase in mitosis. This is the phase after metaphase 2, wherein the remaining centromeric cohesins, which are not protected by the Gaussian, are cleaved. This allows the sister chromatids to separate and move to the opposite poles of the cell. As you can see here, the centromeres divide and the sister chromatids move to the opposite sides of the cell. And the last phase is telophase 2. Similarly to telophase 1, chromosomes arrive at the poles of the cell and decondense. Nuclear envelopes surround the 4 nuclei, and then cytokinesis follows, dividing the cytoplasm of the existing 2 cells. And that's it! That ends our episode about meiosis. Thank you for watching and see you next time!
Moving on to the next topics general biology 1 has to offer, we were challenged to present the meiosis process of cell division. However, typical presentation was not suggested. Instead, we students were tasked to build a model of the aforementioned. Hence, we Team C did our very best as acceptance to this challenge. The project work plan, as decided by the leader, picks sub-teams that have different roles for the model construction, for the video presentation, and for the reflection writing. Our team agreed on the plan where a sub-team will produce the model and will hand the latter afterwards to every assigned presenter. So, for the meiosis model, all recycled materials were allowed to be utilized. Our team considered using plastic straws, paper plates, nylon cable ties, and rubbers. We then produced a meiosis model with quality, which received a big thumbs up from every team member. The 5 presenters who were tasked and divided to report the processes happening in each stage were handed with the Meiosis stage model after it has been built, of course with consideration of the pandemic health protocols. The said presenters then compiled the recorded explanation of each stage Meiosis has as one through a creative manner. It did not end here, as before turning in our output, our team watched the said video so that we simultaneously learn and be more knowledgeable about meiosis. With the great teamwork we possessed throughout the process, we have given ourselves new insights and academic fulfillment.